Hi, my name is Mika Matera Vatnick, and I'm a fourth year MD PhD student at the University of California, San Francisco, or UCSF. I'm completing my thesis work with Dr. Peter Turnbaugh, where I'm studying how the trillions of bacteria living in your intestine, called the gut microbiome, influences the efficacy of GLP-1 receptor agonists, drugs or obesity treatments like Ozempic. So the title of my project is Primed or Unprimed Baseline Microbiome as a Predictor of GLP-1 Receptor Agonist Efficacy. So you might have heard of Ozempic as a miracle drug that has transformed the treatment of obesity and diabetes. But one thing that you might not have heard is that GLP-1 receptor agonists work better in some patients over others. This is important because 1 in 8 Americans as of 2025 have been on GLP-1 receptor agonists, and 1/3 of these patients don't exhibit clinically meaningful weight loss. What factors, whether in the human or in the gut microbiome, explain this interindividual variability in efficacy is poorly understood. And so the goal of this project is to identify a responder-associated signature or a non-responder-associated signature that we can understand and later exploit to make all patients respond to GLP-1 receptor agonists. So in order to do this, we teamed up with our colleagues at the UCSF Department of Endocrinology to sequence the stool of patients initiating treatment on GLP-1 receptor agonists. And so we collected stool at baseline, 2 weeks, 3 months, and 6 months. And today I'll be focusing on mainly the baseline stool. Now, metagenomic sequencing is a very common technique that we use in the gut microbiome field in order to understand what bacteria are there and what are the genes that the bacteria have, which gives us an insight into what the bacteria may be doing. How do we define response to GLP-1 receptor agonists, specifically in weight loss, is as 5% or more body weight loss in 3 to 6 months. Here we see the distribution of body weight loss of our patients, which shows immense variation. And in gray are the non-responders who don't reach the 5%, and in blue are the patients who achieve 5% weight loss. Now, does the baseline gut microbiome predict response to GLP-1 receptor agonists? I'm going to give you a flash explanation as to what we found up to this point, which is that patients who have a baseline gut microbiome that has higher richness or higher diversity, as measured by Shannon diversity, seem to exhibit greater weight loss at 6 months. And so greater richness, more weight loss. We also found that the baseline microbiome composition differs by responder status to GLP-1 receptor agonists, as shown by the separation between the red dots and the blue dots on this plot. We also have found that there are particular gut microbes at baseline that associate with greater or less weight loss at 6 months. In blue are the microbes that associate with greater weight loss, and in red are the microbes that associate with lower weight loss. And so the takeaways are that responders have higher microbiome diversity at baseline, that the baseline gut microbiome composition is different between responders and non-responders. And we're currently really excited about some hits that we're following up with lots of experiments to try to understand the mechanism through which these microbes may be mediating the efficacy of GLP-1 receptor agonists, with the objective of increasing efficacy for everyone. And I just wanted to thank the ARCS Foundation for their continued support, without which this work would not be possible.